asylum in Frankfurt, a German physician by the name of Alzheimer identified a patient that had a weird kind of dementia. This patient had severe memory deficits, but it didn't seem to affect the other faculties in her brain. He observed this patient for 5 years, and he saw these deficits leak out from the memory centers of the brain and eventually take her personality, take her ability to talk, take her ability to move, and eventually she died of frailty in 1906. Alois Alzheimer then did an autopsy and carefully took sections of her brain to look for any anomalies in the brain that might explain the symptoms that he was seeing. He saw 3 anomalies: giant protein aggregates, insoluble aggregates in the tissue of the brain. He used a microscope to look closely at the brain cells, and in the brain cells he saw tiny tangles of proteins within the cells themselves. And the third feature of Alzheimer's disease, clusters of cells that he described as non-neuronal cells that didn't look like they should be in the brain. They're not normally there, these clusters. Now, researchers ever since Alois Alzheimer's have been fascinated by what's causing this disease and trying to cure this disease. And for the better part of the last 100 years, our research has focused on those first 2 anomalies: the protein aggregates in the tissue of the brain and the protein tangles within themselves. We've performed numerous clinical trials that target these proteins with no success. And we now look in the brains of healthy patients and we see these same protein aggregates. And what we're now starting to realize, perhaps it was that third feature that Alois Alzheimer's described that was crucial to the disease. Perhaps you needed all 3 protein aggregates outside, inside the cell, and those clusters of non-neuronal cells. We now know what those clusters of non-neuronal cells are, and it was inflammation. It was your immune cells reacting inside the brain to cause inflammation. Now, what is inflammation? Well, we've all had the swollen ankle from spraining our ankle. We've all had swelling from spraining our ankle. That's inflammation. It's your body's response to tissue damage or infection. Immune cells from around your body will be recruiting into the tissue. The large immune cells will begin to eat the damaged tissue. The small immune cells start to release toxic chemicals to clean up the area, including hypochlorite, which is chemically identical to household bleach. So your immune system is producing bleach for the very same reason we use bleach. It sterilizes the area. But if this is chronic or excessive, the large immune cells can start to eat healthy cells and healthy structures. The small immune cells can produce too much toxic compounds over too long a period and start to cause damage in these healthy parts of the brain, in the healthy parts of the tissue. Now think about that in the context of your brain. In Alzheimer's disease, your brain is inflamed. Think about that. But inflammation can also be good. It initiates the repair processes after tissue damage. And this is the double-edged sword of inflammation. Now, my entire career has researched what molecular switches can we switch off and switch on in these immune cells to push them towards that repairing response and away from that more damaging response, producing those toxic compounds, for example. My career started in New Zealand, which Mick Jagger described as the arsehole of the world. It's a beautiful arsehole. And it has some fantastic universities, but I wanted to research at a world-leading research centre. So I looked to the University of Manchester, who is the #1 university for inflammation in the brain. In particular, Manchester is focused on this one molecular switch that pushes the immune cells to that damaging type, and it's called IL-1. Now, this research was pioneered by Dame Nancy Rothwell. Her lab set up this research in IL-1. She's a personal hero, and I must admit, a slight crush. Now, my research is building on this IL-1 molecular switch to research it in Alzheimer's disease. Is this the switch that is causing damaging inflammation in the brains of Alzheimer's disease? To do this, I needed a drug that would inhibit this switch. So in cells in a dish, I stimulated them to make them inflamed. And then I screened hundreds upon hundreds of drugs to see if I could flick off this molecular switch. And fortunately, I found a small group of pain relievers that happened to switch off the IL-1 signal, that inflammatory switch, too. So now that I had this drug, I could now take it into animal models. Unfortunately, this is the only way to take it to this next step. Now, we had mice that we've genetically inserted a gene that we know causes Alzheimer's disease. And these mice develop Alzheimer's disease. To one half of these mice, I administered this drug that flips off the IL-1 switch. To the other half, I gave a placebo. I tracked their memory using maze tasks. How good are they at learning a maze? And what I found was the mice on the drugs did not have any memory decline throughout their life, whereas the mice on the placebo-controlled drugs declined dramatically. I then, just like Eloise Alzheimer's, carefully sectioned the brains of the mice after doing an autopsy and looked for those anomalies in the brain. In the drug-treated mice, we saw no cell clusters. There was no inflammation going on in the brains of these mice. This took me to the next step of this research, because what was cool is I was looking at existing drugs that people are taking right now. So I could look at a clinical data set that was kept in America and Australia. They closely followed the cognitive decline of an aging population. And I used advanced statistical techniques to pick out the cognitive decline in the patients who happened to be taking the pain-relieving drugs that flipped off the IL-1 switch, off that inflammatory switch. And I could compare it to other patients that were taking other pain-relieving drugs. That way I'm comparing apples with apples. And I saw a 45% reduction in decline in the patients that were taking the drugs that flipped off the IL-1 switch, that flipped off the inflammatory switch. So it was at Manchester I had the opportunity to follow my research from cells to animals and into a clinical data set. Now I've been asked to do a call to action. There are 675,000 people in the UK with Alzheimer's disease right now. And that comes at a huge cost. $27 billion a year to care for these patients. But also, I believe it comes at a greater social cost to the families of those patients and the people suffering the diseases themselves. Yet we only put $85 million a year towards research. That's 0.3% of an investment to stop this $27 billion a year problem. And I would like to compare this to cancer. Cancer gets 5 times the amount of research funding as Alzheimer's disease. We have no treatments in Alzheimer's disease in the clinic. Cancer has hundreds. We're making huge gains. One example is, if you're taking the leading drugs in lung cancer right now, you are 6-fold more likely to survive 10 years than you would be if you weren't taking those drugs. We're making massive gains in cancer. I could say the same about HIV, which people are now living as long as people who are HIV negative on these new drugs that have been discovered by research. I could also say the same thing about blood vessel diseases. Heart attacks and strokes are declining because we've got modern drugs that can control blood pressure and cholesterol. Research gets results. And so what I ask is you do what you can to give Alzheimer's researchers the same opportunities that cancer researchers are getting today. Thank you.